All right, ladies and gentlemen, welcome to our discussion on chapter 4, where we're going to go into the information about carbon and a lot of the stuff that we actually talked about already in our class about carbon and how carbon interacts. One of the bigger things about understanding organic chemistry in general is that we have to understand that when we're talking organic chemistry, what we're saying is that it's the study of carbon. So we're dealing with carbon in this case. A lot of the information is just dealing with carbon. So what we're trying to do is look at this on a bigger scale. What does it mean to be organic? Well, it means that we're dealing with carbon. So what if I wrote down a couple examples? What if I said H2O, CaCl, NaOH, and CH4? What are what would this be? What when I'm saying this, which one of these would actually be considered organic? Well, the answer to that is CH4. And the reason is because of that carbon. Nitrogen, oxygen, hydrogen in NaOH, calcium and chlorine in CaCl, hydrogen and two hydrogens and an oxygen in H2O. The only one that's really organic is considered organic because it's carbon based is CH4. When we look at this, we know our most common of these elements, carbon, hydrogen, oxygen, nitrogen. That was our four most common, right? Anything else we consider to be, it's going to be considered minute or a trace element. So with this carbon, hydrogen, oxygen, nitrogen, there were some sulfur, some phosphorus that comes into play, but anything else, all else would be considered trace elements. Now, the thing is that carbon itself has the ability to make a covalent bond with carbon, and then carbon, and then carbon, and then carbon, and create this long chain of carbons. And let's say hydrogens were attaching on, we'd have this kind of long chain that could go on technically over and over and over and over again, because as long as we follow our rules of bonding, this carbon has four spots where these hydrogens can attach on. So when we're talking about this, when we look at the electron configuration of carbon, it's important to realize that carbon is going to have four electrons in its outer shell. With having four electrons, it means that there's four spots. One, two, three, four spots where other electrons can come in and they could try to go and share. So we say that it has four valence electrons and it can form four covalent bonds. That leads to a valence number of 4. So that means 4 bonds can go. So that makes us have the ability to create these really large complexes. Whether I was talking about that CH4 that we were talking about before, carbon and 4 hydrogens on either side, or we're talking about that larger molecule that I was showing before with, look at something like C6H12O6, which is going to be a sugar, C6H12O6. No matter what, they're combined with carbons. So when we look at these different types of organic molecules, it's important to realize a couple of things. Number one, like the CH4 I was just drawing, this is the carbon with 4 hydrogens. But we have this ball and stick model. If you remember going over in lab, we went over this kind of model. We showed that model. And the space saving model also, or the space filling model, we use that multiple ways too. I use that with like water to show you how the structure of H2O is. But the molecular formula and the structural formula are two different things. So that's kind of something that we want to focus it on. The molecular formula will always tell us the amount of these particular elements in each side. So when it says C2, it means that there's 2 carbons. And when it says H6, it means it's 6 hydrogens. So when we look, 1, 2 carbons, 1, 2, 3, 4, 6 hydrogens all coming together. See that same thing over here where each line is actually going to represent 2 electrons. So each line equals 2 electrons. Over here we have C2H4. Now, the way that that's going to work, sometimes you're like, well, if we have only 2 carbons, how would that work? You know, we had 2 carbons over there, but I needed 6 hydrogens. Well, if there's 4 hydrogens over here, that means there was the presence of something called a double bond. Because if you remember the double bond right over here, 2 sticks, that's actually going to share 4 electrons rather than the 2 electrons that we saw. Now, I'm not going to give you the molecular formula, and you have to give me the structural formula, anything like that. I would want you to look at a structural formula. Words aren't working. My structural formula, and be able to understand the molecular formula that comes off of it. When we talk about these organics and we go over the different functional groups, it's important for you to see those in a structural formula and be able to identify. So that's something that we will talk about. Now, similar to what I was bringing over before, when we say there's unpaired electrons, what that means is that the carbon itself has these electrons that have possible spots for the to attach on. So there's one spot here, there's one spot there, there's one spot there, there's one spot there. So we say that it has a valence. The valence, or the number of covalent bonds that it can form, is going to be a valence number of 4. Now, when we look at that particular configuration, that makes it super easy for things like carbon, hydrogen, oxygen, nitrogen to want to attach on to an additional carbon. So you take a look, hydrogen. When we look at something like hydrogen, this has 1 electron in its outer shell. So that means there's 1 spot open. So there's 1 electron that's needed, so the valence number is 1. In something like oxygen, there's going to be these 6 electrons. So that means there's 1, 2 spots. So there's 2 electrons needed, so the valence number is 2. In nitrogen, we're going to have 5 electrons. So there's 1, 2, 3 spots. So that means the valence number is 3. And in carbon, as we saw, 4 electrons, 4 spots. So 4 electrons needed, the valence number is 4. Okay, so when we're looking at this, it's important for us to realize that what we're seeing with these different carbons, carbon can be single bonded like that, carbon can be double bonded like that. So in the case of CO2, it actually looks like this, carbon double bonded with 2 oxygens like this. And the meaning behind that is because if you look at oxygen, there's 1, 2, 3, 4, and then there's 2 more electrons. Now, these 2 electrons from oxygen and the 2 electrons from carbon can share. And since there's 4 electrons being shared, we get a double bond. On the same thing, 1, 2, 3, 4, the 5 and 6 is going to get shared right over here with this carbon and create another double bond. So you have a double bond here, a double bond here, which gets us CO2. Now, when we look at this carbon, everything that we're seeing about our organics having that carbon as that kind of middle ground, that's going to be your spot that's in the We would call it the backbone. But that backbone, other things can come off of it, like this guy over here, and this guy over here, and this addition right here. The big key portion is having that carbon as the backbone. So it could look like this, it could look like this. Technically, when you look at a ring like this, what we're actually seeing is that each of these lines, as it's coming together, is really going to be our carbons attaching on. So a carbon ring, we'll call it. So you have this carbon ring as opposed to being kind of one of those particular skeletons. Now, when we're looking at these chains have different variations. I definitely would want you to know the variations. Definitely want you to know the variations in these skeletons because it's going to be helpful for identifying things. So number one way that it can change is pure length. If you notice, this is going to be C2, H6. So there's two carbons here, where this is C3, H8, 3 versus 2. That's one way we can see these carbon skeletons kind of vary. We said that already. That's kind of like what I said from the get. But the second part ends up being our branching. If you notice, this is C4, H10. But this is C4, H10. They're both the same, except this carbon looks like this, and this carbon comes out like that. That one branching changes the structure. And if you remember, when we say, if you change the structure, you change the function. That's going to be a key that we see throughout. How about double bonding? Well, not just double bonding, but the positioning of the double bond. You see that double bond right there? Notice how it's in the middle here. By just doing that, it changes the actual form. If it changes the form, it could change the function. And lastly, the presence of rings, like I was showing you before. Something like this is just talking about the carbons that are attached on. But the difference between this ring and this ring is this guy right there. You see that right there? That's actually the presence of your double bond. If you notice, there's two lines there. That's the double bond. Here, there's only single lines. So that means there's only single bonds. When we look at something like a hydrocarbon, the word hydrocarbon refers to carbon and hydrogen. In these cases, you'll see things like fats as being a good hydrocarbon. We have a bunch of different organics, but if you take a look at fats, what they do is they store energy for us. So a fat molecule itself, what it is, it's composed of carbon, hydrogen, and oxygen that's going to store energy for us to use as what I like to call the rainy day fund. The thing about hydrocarbons is that they can release a large amount of energy. They harness a lot of energy and they're combustible. So we use these hydrocarbons in different things, like our fossil fuels are examples of hydrocarbons. Now, similar to when we talked about our different types of elements and how there's different variations that can happen, we see that there is this concept of an isomer. So if you remember, we talked about an element, and then there's isotopes of those elements, and then we talked about ions, right, that are associated. Well, with this, an isomer is going to be a kind of like a compound, a bigger structure, but it has different properties. Like the structural isomer is pretty simple. You take a look at one, and the structure of that other one is different. So remember we talked about branching, right? Branching happens here. Basically, what you're saying with isomers, these two have the same exact breakdown. So this is C5, H12, but this is also C5H12. Their molecular formula is the same, but their structure is different, so they're considered isomers. Next, we have what we call a cis-trans isomer. The cis-trans isomer means if you look at both of these, they're going to be pretty much the same. They both have a double bonded carbon there. They have hydrogens on two of those legs and X's on the other two legs. That could be anything. Now, what they're saying is if you look at these cis-trans isomers, if you have the two X's on the same side, we call that a cis isomer, cis same. If they're on different sides, it's considered a trans isomer. Trans isomer is two opposite sides. Cis isomer is going to be on the same side. And then finally, we have enantiomers. I like to always think of it like your hands. You have your right hand and your left hand. If you are a righty, you do things well with your right hand. Your left hand, it can do the same things, you just don't practice with it as much, so it doesn't come out as great. So what's happening here with these enantiomers, they are mirror images of each other. Now, the thing that happens with mirror images is that one can be perfectly working and the other can be completely non-functional. And that's how this enantiomer makes things a little bit more complicated. Like, look at this. We have ibuprofen. Ibuprofen, as its S-ibuprofen, is going to be effective in reducing inflammation and in pain. But you get the R-ibuprofen, which is just the enantiomer of that, completely ineffective. Same thing with like albuterol, used for like asthma. Relaxes your bronchial airways, improves airflow, right? R-albuterol works out perfect, but the S-albuterol, ineffective. Different structure leads to different functions. That's actually going to be our key. Now, with each of these particular chemicals, what we start seeing is that we have these chemical groups, or what we like to call functional groups. Functional groups end up being what make each of these compounds different. Because technically, I could just have a chain of carbons, but the question is, what's attaching on to that carbon? Now, I'm just making a fictitious kind of connection, but if I just made something like this, the question is, what is this and this? Because depending on what those particular functional groups are, that will change the structure, which will change the function. So we look at different types of classes, look at something like estrogen and testosterone. Estradiol is a type of estrogen. Notice structurally they look very similar, but what's the difference is these blue guys over there. Those are the functional groups. Depending on the functional groups, we're going to see that we have different types of functions. So these are the functional groups that we are going to talk about. Hydroxyl, carbonyl, carboxyl, amino, sulfhydryl, phosphate, and methyl. Those are going to be our functional groups. Now, we should remember this from our lab, but if not, let's go ahead and talk about these. The only one that we kind of added here was this methyl group, and it's actually pretty easy to locate. So first, we'll look at our hydroxyl group. Hydroxyl, you break it down, hydrogen, oxygen. Basically, it's going to either be written out, this line over here represents what's connecting to the carbon of that larger compound, and as that, or like this. Either way, it's going to show up like that, and that is going to give us that alcohol kind of compound name of our hydroxyl. In the ketone and aldehyde, these are going to be different carbonyl groups. Now, what we should realize for both of these, going to be an oxygen double bonded to carbon and two legs coming out. But the question is, what happens after those two legs? Because we have one that looks like this, that has a hydrogen on one of those legs, and one that has nothing. Well, the ketone, I like to think ketone alone, okay, ketone alone, that's going to be by itself. And then the aldehyde, think like the H in hide and the H in hydrogen, aldehyde, is going to be what we see. So over here, this is going to be a ketone alone. And over here, that's an aldehyde with the H on the side. For carboxyl, the thing about this, we have our oxygen and we have our carbon, and the two legs. Notice that between carbonyl and carboxyl groups, they look the same. But the question is, what's attached onto the legs? If we have the legs and attaches onto OH, that is a carboxyl. And the oxyl, for me, I always refer back to the hydroxyl aspect of it. So it has a hydroxyl attached on. So you'll see it like this. The amino group is interesting because I always say it looks like a flux capacitor. It has hydrogen, hydrogen, looking like that, right? With this, you're looking for the nitrogen and two hydrogens attached on. The flux capacitor. It's called the amine or amino group. Sulfhydryl, super easy. That one's just like our hydroxyl, either like that or like that, depending on what side it's going to be on. This is on the right-hand side, so we see the sulfhydryl. Our phosphate, the one thing that I like to think about with a phosphate is that it's going to be our phosphorus with a lot of oxygens on the outside. So look for that phosphorus in the middle. When you see that phosphorus in the middle, that'll give you the key to being a phosphate. And again, the only one that we didn't do is this CH3, which can be led by itself, looking like this, or it would be carbon with three hydrogens. And we'd see something like that. Awesome. The last thing that we would talk about here is the understanding of something called ATP. So our energy source is very important. Remember that we don't create or destroy energy. We can only transfer or transform that energy. So one of the things that we like to do is, as human beings and animals, we like to eat food, and when we eat food, we take in those sugars, and those sugars, the the energy that was formed in the bonds of those sugars, we're going to go ahead and break down those bonds and use that to create an energy source that we can use. And bringing into it, it's called adenosine triphosphate. Remember that phosphate we were talking about? Well, one, two, three of those phosphates put together. So it's an adenosine and three phosphates. In our book, normally what we tend to see is that the ATP is going to be written out like this. So it looks like kind of like a character in a comic book decided to hit somebody. So I always think of it like it's like bam, kerplow, splat, right? Those kind of words. So I'm always like bam, ATP. So in order for us to go and utilize the kind of energy that we want to use, we have our potential to break down this energy to go break down this energy source to go and release the energy that we need. There's a concept. Life requires work, but work requires energy. So everything that we do to create energy is so that we can live our life. So again, I shouldn't say create the energy, but create our sources of energy. is to go and have that life, have our life. So we're going to go and uh, transform the energy the way that we can. And ATP is our best source. And what happens here is I have ATP, right, which is adenosine triphosphate. And what ends up happening with this whole process is that it's going to react with water. Just as a side note, H2O, if I add H2O, I break a bond. Think like when you're eating food and your saliva has a lot of water in it. If you add water, you break a bond. We call that hydrolysis. If I subtract water, if I take away water, I'm actually going to make a bond. We call that dehydration synthesis. So in this case, I'm reacting with water to add water, and I'm breaking it down into ADP and inorganic phosphate and that energy. So as kind of like a little, I like this little flow chart to do. So I'll take ATP and I'll stay true to our text and have our BAM ATP, and you add water. And when you do that, you're going to get ADP and our inorganic phosphate. But if I added those together and pulled out the water, I'd make ATP, right? So there's this kind of concept, there's this flow that happens. The thing is, in order for us to make our ATP, we have to input energy. But when we break the ATP, what we're going to see is that we output energy. So this kind of energy can come from the food that we eat. This energy is the energy that we need to do work. So when we talk about ATP, what runs everything is really the hydrolysis of ATP. What actually runs everything is the hydrolysis of ATP. So testing our understanding by looking at something like this, that right there, what is that? Good, that's a hydroxyl group. Over here, what is this? Good, that's an amino group. And one over here, good, that's a carboxyl group. We look at this over here, these are both carbonyls. So this one right there, that's a ketone alone. And then over here, this is going to be an aldehyde because of that hydrogen. If we look at something like this, A, that's going to contain an aldehyde. B contains a hydroxyl. D is going to be a methyl group. C, you know, in this case, what they're looking for, probably just that single bond, showing that single bond that's there. This example of L-DOPA and D-DOPA is just showing that they are two enantiomers. The enantiomers are actually going to be mirror images of each other. Awesome. And that was chapter 4.